Artificial Intelligence Dr. www.abcpharma.net/English. User question: what are the specific molecular mechanisms linking mitochondrial dysfunction to impaired insulin signaling pathways, and how does this contribute to the development and progression of insulin resistance? Claude AI answer: this is an excellent and complex question that delves into the heart of the relationship between mitochondrial function and insulin resistance. Let's break down the specific molecular mechanisms linking these two processes. 1. Oxidative stress and ROS production. Mitochondrial dysfunction often leads to increased production of reactive oxygen species, ROS. Excessive ROS can activate stress-sensitive kinases such as C-jun N-terminal kinase, JNK, I-kappa-B kinase, IKK, and protein kinase C, PKC, isoforms. These kinases phosphorylate insulin receptor substrate, IRS, proteins on serine residues, inhibiting their tyrosine phosphorylation and disrupting insulin signaling. ROS can also directly oxidize and inactivate key components of the insulin signaling pathway, including the insulin receptor and glucose transporter 4, GLUT4. 2. Altered cellular energy status. Mitochondrial dysfunction can lead to reduced ATP production and an increase in the AMP-ATP ratio. This activates AMP-activated protein kinase, AMPK, which can have both insulin-sensitizing and insulin-desensitizing effects depending on the cellular context and duration of activation. Chronic AMPK activation can lead to increased lipid accumulation and insulin resistance in some tissues. 3. Impaired fatty acid oxidation. Mitochondrial dysfunction often results in reduced capacity for fatty acid oxidation. This leads to accumulation of lipid intermediates such as diacylglycerols, DAGs, and ceramides. DAGs activate PKC isoforms, which phosphorylate IRS proteins on inhibitory serine residues. Ceramides activate protein phosphatase 2A, PP2A, which dephosphorylates and inactivates ACT protein kinase B, a key mediator of insulin signaling. 4. Mitochondrial unfolded protein response, UPRMT. Mitochondrial stress can trigger the UPRMT, leading to activation of transcription factors like CHOP and ATF5. These factors can induce expression of genes that interfere with insulin signaling or promote inflammation. 5. Altered calcium homeostasis. Mitochondrial dysfunction can disrupt cellular calcium homeostasis. Changes in cytosolic calcium levels can affect insulin secretion in pancreatic beta cells and insulin signaling in target tissues. Calcium overload can activate calcium-dependent proteases that degrade key insulin signaling proteins. 6. Inflammation. Dysfunctional mitochondria can release damage-associated molecular patterns, DAMPs, like mtDNA and formal peptides. These DAMPs activate inflammatory pathways, including the NLRP3 inflammasome. Chronic low-grade inflammation contributes to insulin resistance through various mechanisms, including activation of JNK and IKK. 7. Mitochondrial dynamics. Impaired mitochondrial fusion and fission processes can affect mitochondrial quality control and function. Alterations in key proteins like mitofusin 2, MFN2, have been linked to insulin resistance. Disrupted mitochondrial dynamics can affect mitochondrial distribution and function in insulin-sensitive tissues. 8. Impaired mitochondrial biogenesis. Reduced activity of PGC1-alpha, a key regulator of mitochondrial biogenesis, is associated with insulin resistance. Decreased mitochondrial content and function in insulin-sensitive tissues contribute to impaired glucose uptake and utilization. 9. Altered metabolite profiles. Mitochondrial dysfunction can lead to accumulation of metabolites like acyl carnitines and branched-chain amino acids. These metabolites can interfere with insulin signaling and contribute to insulin resistance. 10. Epigenetic changes. Mitochondrial dysfunction can lead to alterations in the cellular redox state and metabolite levels. These changes can affect epigenetic modifications, leading to altered expression of genes involved in insulin signaling and glucose metabolism. The development and progression of insulin resistance due to these mechanisms is often a vicious cycle. Initial mitochondrial dysfunction leads to impaired insulin signaling, which can further exacerbate mitochondrial dysfunction through reduced glucose uptake and altered cellular metabolism. Understanding these specific molecular mechanisms provides potential targets for therapeutic interventions. Strategies aimed at improving mitochondrial function, reducing oxidative stress, enhancing fatty acid oxidation, and modulating mitochondrial dynamics could potentially improve insulin sensitivity and prevent or treat insulin resistance. It's important to note that the relative contribution of these mechanisms may vary depending on the tissue type, the underlying cause of mitochondrial dysfunction, and the stage of insulin resistance development. Future research will likely uncover additional mechanisms and refine our understanding of these complex interactions.